Welcome to lecture today. Today we're going to talk about membranes and all the fun associated with membranes. It's a fairly substantial topic. Unfortunately, I don't think we're going to do it justice. It is one of the longer topics. I think the important things you get out of this is the fluid mosaic model and what that means and what that does. Quick case study for you, just to kind of put a frame of reference. Edward is a 35-year-old white male. He came to a neurological clinic for evaluation of his long-term neurological complaints, noting that for many years he has noticed some significant changes in his neurological function, particularly heat intolerance, precipitating a stumbling gait and a tendency to fall. His visual acuity also seemed to change periodically during the several preceding years. 2 months ago, the patient Edward was working very hard and was under a lot of stress. He got sick with a flu and his neurological condition worsened. At that time, he could just not hold objects in his hand. He had significant tremors and had suffered severe exhaustion. He also had several bad falls. What do you think? Some more information. What do you think it is? Spinal tap was done, which revealed the presence of oligoclonal bands in the CSF. A neurological exam was conducted using a fundoscope. Visual evoked response testing was abnormal, slowed conduction in the optic nerve. MRI scan was performed at that time and revealed multifocal white matter disease areas of increased T2 signal in both cerebral hemispheres. If you want to pause now, you can look it up and see what you think it is. Here's what oligoclonal bands in the CSF look like. You will note that this is a gel electrophoresis of proteins that's been Kumasi stained. It's an SDS page gel, and they just take the spinal fluid out and run it on a gel. The answer to the question is multiple sclerosis. It's an autoimmune disease that affects the central nervous system, and we'll talk about it because it primarily affects membranes. You can get MS at any age. There's greater than 400,000 cases in the United States, 2.5 million worldwide, 200 new cases each week. If you live up north in our area of the woods, you have a higher prevalence of getting it. The ratio of women and men with MS is 2 to 1. 2 women for every 1 man. There's a genetic component. Identical twins raises the risk. People with MS also are slightly increased risk of developing MS if you have type 1 diabetes, thyroid disease, or inflammatory bowel disease. All those, notably, could be associated with autoimmune. So the cost is $8,000 to $54,000 per patient per year in the United States. What happens? In a normal nerve cell, your axon, you have a myelin sheath around that axon. That's the way the information is conducted. Electrical current is conducted down. The myelin sheath provides insulation. When it gets damaged and scarred, usually because of an autoimmune disease, you get significant problems. And this is a membrane problem. This is why we're talking about it. So let's jump into the topic again. Structural lipids and membranes. We've talked about this in the previous lectures, but I want to just reiterate a couple points. The structural lipids make up membranes, and there's usually a polar head group with non-polar tail. Usually that's a fatty acid. Diversification can come when we modify the different backbone of the fatty acid. We could change the fatty acid themselves, or we can modify the head groups. Again, different organisms have different membrane lipid head group compositions, and different tissues have different membrane lipid head group compositions, which is a topic we'll discuss today in some detail. First and foremost, lipids aggregate into different structures in water, and the 2 primary are micelles and vesicles. Micelles are basically a single layer where all of the fatty acids go into the center. It forms a sphere, and the polar head groups are out into the world, out into the aqueous environment. Individual web-shaped cross-sectional head groups, because they're bigger, will form these. So usually the structure's dependent upon the type of lipid, the concentration, and whether they're going to form into bilayers. So forms of the solution, if you have an amphipathic, meaning you have a polar and a nonpolar molecule, in this situation you have a polar head group. that will cause these structures to form. You can also get it if you have SDS in the solution, and a micelle can be as few as a few thousand lipid molecules. A few other points. Micelles, again, are sequestered at the core. There's virtually no water in that hydrophobic interior. When you start getting into other liquid aggregations, such as the vesicle, there's a bilayer that forms. It's a closed bilayer, and you can get an aqueous environment in the core inside that. So you have a polar head group on the inside, you have a hydrophobic bilayer that's formed, and then on the outside you have polar head group. So it's hollow. It can be a liposome, and that forms inside them. You can seal off a aqueous core. They're very useful for drug carriers. If you're going to start to see this, this is one of the reasons why I'm talking about it. You're going to see this useful as we get forward as a way of increasing and changing the PK/PD profiles of drugs, as well as deliver them to places that are difficult to deliver to. In addition, you're going to start seeing these in multiple other places, because vesicles can fuse readily with cell membranes and with each other based on the properties of the membranes. And so you're going to see these as drug carriers. You're going to see these as ways to attack disease states, deliver DNA, all sorts of fun stuff. So let's dive into a little bit about the membrane bilayer. Individual units if it forms a bilayer are cylindrical, as you might recall that they're wedged. In other words, the polar head groups are very large. So if they're cylindrical, the cross-sectional area of the head equals that of the side chain. You're going to form a bilayer. Again, we have a polar head and a non-polar tail. The hydrophobic head interacts with water. The hydrophobic fatty acid chains are packed on the inside. One leaf faces the cytoplasm, and another leaf faces the extracellular matrix. There are complex structures that can form with these pliable sheets, and in a normal membrane of a cell, in any membrane of a cell, there's a variety of lipids and proteins. Some membrane and lipid are glycosylated. Basically, these are separation mechanisms, and there's different ways to provide that interface in that bilayer. There's different ways that these bilayers can interface with the inside and exterior space, and these are, as such, these are commonly asymmetrical bilayers, meaning one layer is different in composition than the other. This is what an electron micrograph looks at. This is an endoplasmic reticulum, and you can see the nuclear pore, the mitochondria, and you can see secretory granules. And so it shows, I think it's really an interesting, it shows all the different lipid compositions. EM is going to provide increasingly important information on these kind of structures and also viruses. So what are the functions of a membrane? A membrane is composed of multiple different entities, including proteins, oligosaccharides, glycolipids, phospholipids, cholesterols in there, peripheral proteins. You have all kinds of stuff. Basically, membranes function to separate out biomolecules from each other. It sets a boundary for the cell. It allows for import and export. It allows for increased concentrations. We can import nutrients, export waste and toxins. We can retain metabolites and ions within the cell. We can sense external signals and transmit information into the cell, all via the membrane. The membrane provides membranes of all kinds can provide compartmentalization of the cell to separate things like energy production and reaction specific opportunities. We can keep proteolytic enzymes away from important cellular functions. We can produce and transmit nerve cells. I mean, the list is amazing what we can do. We can support synthesis of ATP, store energy, transmit nerve signals, on and on and on across a membrane. Membrane serves as two important gradients in life. First off, we have an electrical gradient across, meaning we have a potential across. Usually this is maintained by protons. That's the electrical component, but we also have a chemical gradient where we keep nutrients in and out, and those have numerous functionalities within a cell, as we've mentioned. Common features of membranes. Membranes are about 3 to 10 nanometers thick, or 30 to 100 angstroms. There's a main structure, which is a bilayer. There's two leaves of that membrane. There's an exception. Archaeobacteria have a monolayer of bifunctional lipids. They form spontaneously in aqueous solution, and they're stabilized by non-covalent forces, primarily the hydrophobic effect, but you also see van der Waals forces in there as they interact. Protein molecules can span the lipid bilayer. Important point here is they are very asymmetrical. Some lipids are found preferably on the inside and some on the outside, as we mentioned earlier. Carbohydrate moieties are always on the outside of a cell, and I say always very hesitantly, because I'm sure there's an exception to that, but generally, in 99.99% of the cases, carbohydrate moieties are on the outside, and the potential relative from the inside to the outside is negative 60 millivolts. They are very fluid structures. They're two-dimensional solutions of oriented lipids. Those are the common features of membranes. Now, in 1972, Singer and Nicholson at UCSD proposed what is called the fluid mosaic model of membranes. This is the topic you'll want to know. Lipids form a viscous two-dimensional solvent in which proteins are inserted and integrated more or less deeply. Integral proteins are firmly associated with the membrane and often span the bilayer. Peripheral proteins are weakly associated and can be removed easily. Most are non-covalently attached. Most are, I shouldn't say most, but some are linked to lipid membranes. Okay, so this is what it looks like. We have all kinds, so there's a basic lipid bilayer, okay? On the outside, you're going to have carbohydrate entities, glycolipids, oligosaccharides off of glycoproteins, GPI anchored proteins, and so you have all these things on the outside of a cell. You also have things like pores and pumps, and you also have sterols, including cholesterol. Those cholesterol provide different permeability of the membrane. We have integral proteins, which is a single transmembrane helix, which can provide all sorts of signaling to the cells. We have our phospholipid polar head groups that are on the outside. We have sphingolipids on the inside of membrane. We have peripheral proteins that are associated on the interior matrix, and we have peripheral proteins that are covalently linked to a lipid. We have all kinds of stuff going on in this. The fatty acyl chains in the interior membrane form a fluid hydrophobic region. Integral proteins float in the sea of lipid, and so this hydrophobic interactions kind of keep these molecules, as well as van der Waals forces, kind of keep these things very fluid, and they're free to move laterally. Proteins and lipids are laterally. They can diffuse. They don't flip sides, but they move laterally, and they also aggregate in particular rafts is what the term that we're going to use, and we'll discuss all that. Carbohydrate moieties attach to some proteins and lipids and into the plasma membrane. On the extracellular side, this is the important part of the fluid mosaic model, and I would understand all the different components and the different functions. Membrane composition is highly variable, and so different types of so 30% in the human myelin sheath, 30% protein, 30% phospholipids, 19% sterols. And the sterol type is cholesterol. You see galactolipids and plasmalogens in there. A mouse liver, on the other hand, is 45% proteins, 27% phospholipids, 25% sterol. And so as you go through this, you kind of see how all of this every component is different. And the other important thing that's going to happen is that the inside is not going to equal the outside. The type of phospholipid varies per the abundance of sterols varies, and the type, and you have all different kinds, the protein concentration. So membrane composition is highly variable. Again, this kind of also shows that the inside and the outside of like mitochondria are different. It gives you numbers that kind of show how this works, and the different types have different structures and thus different function. This is a functional specialization of each membrane, and that's dictated by the composition, or vice versa. Cholesterol is predominant in plasma membranes, but barely detectable in things like mitochondrial membranes. Cardiolipid is a major component of intermitochondrial membrane, but not of the plasma membrane. So you can just see all over the place we have different components that are found in different compositions. Again, that structure leads to different functions of those membranes. Again, another important thing, inner and outer membranes are asymmetric. Two leaflets have different lipid composition. The outer leaflet is often more positively charged, so phosphatidylserine as outside has a special, well, we already discussed this. Phosphatidylserine, the outer leaflet is positively charged. Phosphatidylserine on the outside means that apoptosis needs to occur, or cellular damage has occurred. So, I mean, other things, the cellular composition is very, very important, as we've discussed earlier. Again, so things like if you have a transport vesicle, you have single lipids plus cholesterol. And on the outside, on the inside, you have phosphatidylethylamine plus phosphatidylserine. In things like a trans-Golgi network, you have phosphatidylcholine plus sphingolipids plus cholesterol on the outside, whereas on the inside, you have phosphatidylethylamine and phosphatidylserine. On the Golgi, you have phosphatidylethylamine and phosphatidylserine on the outside, and phosphatidylcholine on the inside. Hopefully, you're beginning to catch the drift of how this works. And a charge, structure, charge, all those things play an important role of where they're found, where membranes are, or what composition of membranes occur. So what are the function of proteins in a membrane? We have three main ones. As receptors, they detect signals from the outside, things like light, hormone, neurotransmitters, pheromones, these are all GPCRs, by the way, which we'll talk about again when we talk about biosignaling. These receptors all receive signals from the outside. All of them are GPCRs. We have channel, gates, and pumps. We'll talk about extensively these. So nutrients can get pumped in, and ions can get pumped in and out. Neurotransmitters are also pumped in as a mechanism of sensing. Enzymes are also in the membranes, things like lipid biosynthesis. For example, some acyltransferases, FO1 ATPase is also an enzyme that's found in the membranes, as are numerous others that we will discuss in detail in the coming weeks. Peripheral membrane proteins associated with the polar head group of membranes. So these proteins are transmembrane, so they're going to, and so there's some residues that are going to associate with the polar head groups. They're relatively loosely associated with membranes through ionic interactions with the lipids or aqueous domains of the integral membrane proteins. They can be removed by disrupting them with ionic interactions, either with high salt or a change in pH. So we purify peripheral membranes, and they no longer are associated with lipids. Integral membrane proteins span the entire membrane. They have asymmetry like the membrane, different domains and different compartments. They're tightly associated with the membrane. In particular, the hydrophobic stretches in the protein interacted with that highly hydrophobic region of the membrane where the fatty acids occur. The removal of detergents disrupts. They're removed by detergents. So we put SDS or one of those detergents like that in there, and that's going to pull them out, although they will still have phospholipids associated with them as you purify them. Integral membrane proteins commonly have like a 7 alpha helix domain, again highly hydrophobic residues on the that fit in the outside. This happens to be PDB 2AT9, and you can look into this. It has 7 alpha helices that traverse the membrane. They're cluster. This is a light-absorbing pigment, retinal. It's buried deep. This is a GPCR, by the way. The helices are corresponded to the hydropathy plot that we're going to show a little later, and you can kind of get an idea of which residues are hydrophobic. So, integral membrane proteins, receptors commonly. There are 6 types of these, and you can see them generally here. So type 6 is on the left. We have 1 spanning alpha helix domain. We have a GPI anchored on the cytosolic side, and we have a cytosolic arm, or a arm that goes into the extracellular matrix. So that's type 6. Type 4 is has a lipid anchor, and that on the exterior side of the cell. Type 5 is a 5 member alpha helices. I shouldn't say that. It's not. Type 5 is a alpha helices. Usually they form complex. Type 3 is the one that we're commonly think of that are all connected. that have transmembrane domain, a large number of transmembrane domains that kind of go in and out of the matrix. Type 5 does not. It only, all these are aggregates, are subunits that are interacting with each other. It's a heteropolymer commonly. Type 2, we have amine groups that stick out into the extracellular matrix, and a carboxyl group that's in the interior, and a type 1 is just the opposite of that. So all of these have different functionalities and provide important functionalities. So we can figure out where these transmembrane domains are by just doing a hydropathy plot. That just tells you where it's hydrophobic. And so you can tell that in the middle of some of these, you're going to tell what kind of membrane type is going to interact with that. So it's going to tell you where you're going to have the membranes and what type of membranes, or sorry, it's going to tell you how it's going to interact with the different lipid composition. So it could tell you where the transmembrane domains are going to be. So amino acids, again, this is what I meant to say earlier, I apologize. So amino acids in the membrane proteins tend to cluster on their hydrophobic nature. So transmembrane segments are predominantly hydrophobic. Things like tyrosine and tryptophan tend to cluster at non-polar-polar interfaces. Charged amino acids are generally only found in the aqueous domains. And so if you know the tertiary structure of a molecule, you can kind of get an idea, and you can see where they actually occur. You can see that charged residues are generally on the inside of a protein, or on the outside of the surface of a protein in the extracellular space or in the cytosolic space, but not in the membrane. And then we have right at the interface, we have a lot of tryptophan and tyrosine. This is definitely going to be something that's on the exam, on the exams. There's also beta sheets. Commonly you see a lot of alpha helices. We also have some beta sheets in membrane proteins, and these are all in beta barrel forms, which means that these beta sheets form complex tertiary structures. Some say super secondary structures, but they form, whatever you want to call it, super secondary or tertiary, they form complex structures that span transmembrane. These are all have PDB codes in them. If you look them up, you can kind of pull them into PyMol if you'd like. We do have lipid length membrane proteins. We briefly mentioned these the other in the other lecture. So if you put a palmitoyl group, so if you have a post-translational modification, there's certainly post-translational modifications. If you look at that chart that I've presented the other day, so we can post-translationally modify a protein. Commonly you'll put it on things like a cysteine, or if you have an amine, something like a lysine or an arginine, or sulfur group, so farnesyl will form on a cysteine. The myristoyl group will form on the amino terminal glycine. Palmitoyl groups will form on cysteine or serine, and they anchor the protein into the surface. We also have GPI anchors in there, which we mentioned earlier, where we have a lipid followed by a phosphate linkage into a carbohydrate structure. That carbohydrate structure has another phosphate linkage onto the protein. And so lipids can be anchored into the protein or into the membrane that allow them to be receptors or structural entities. They also can interface with things like cholesterol and other sterols. GPI anchors are generally only found on the outside face of a plasma membrane. That's important to note. All right. So we've talked a lot about integral proteins. So let's talk about what happens in multiple sclerosis. We have a protein, MBP. And MBP and or PLP provide structural integrity, and they allow, in an axon, they allow for insulation. And what you end up happening, and you can look this up, it's a very, very interesting topic on PyMol if you'd like. There are autoantibodies that form against MBP generally. So there's inflammation that attacks these sheets. One of the two things. There are multiple forms of multiple sclerosis. This way is just one. This MBP molecule, which is a structural molecule, you can see that there are 3 membranes in an axon. That forms a myelin sheath. That myelin sheath gets damaged either through high inflammation or autoimmune disease. And MBP is the target. So we've developed natalizumab, which is a humanized monoclonal antibody against alpha-4 integrin. It treats multiple sclerosis and Crohn's disease. Basically, it reduces inflammation of immune cells. And so these integrins, which are transmembrane receptors, are the bridges for the cell-to-cell and cell extracellular matrix interactions. And when it's triggered, chemical pathway to the interior, and those integrins are important in maintaining the normal function of a cell. This helps reduce that. All right. I want to reiterate to you, let's go back. The important part of this lecture is the fluid mosaic model, which basically states that in a plasma membrane structure, there are fatty acid acyl chains in the interior, form a fluid hydrophobic region. Integral proteins float in the sea of lipids. And they're all held together by this hydrophobic interaction with non-polar amino acid side chains. Both the proteins and lipids are free to move laterally. They don't switch between inside and outside, but they do kind of float inside, in this sea of membranes. Carbohydrate moieties are attached on the exterior side of that surface. Okay. Again, cell membranes are asymmetric. I just want to reiterate that point. A couple other points that I want to reiterate. Plasma membranes are dynamic and flexible. They can exist in various phases, and they can undergo phase transitions. They're very not permeable to large polar solutes and ions. They are permeable to small polar solutes and non-polar compounds. So permeability can be artificially increased by chemical treatment, i.e. when we want DNA to go into the cell, we can make that happen by chemical means or by electricity. That's called transforming a cell. So some of the other ways, membranes have phases, and the composition of the cell, this is going to be on your exam, by the way, I can just promise you. So, and on the ACS exam as well. So membrane composition can be dictated by the type. So if there's a gel phase, individual molecules do not move around in a gel phase, but in a fluid state, individual molecules can move around. Heating up can cause a transition from a gel to a fluid. Also, physiological conditions can cause that, but we need fluidity for proper function, and this is where the discussion, this is where the test topics come in. So membrane fluidity is determined mainly by the fatty acid composition. If you want shorter and more unsaturated fatty acid chains will make a membrane more fluid. And so if you recall back to the melting temperature of fatty acids, the shorter and more unsaturated fatty acids have a lower melting temperature and therefore are more fluid. At higher temperatures, cells need more saturated fatty acids to maintain the cell integrity. At lower temperatures, cells need more unsaturated fatty acid to maintain fluidity. So, cholesterol, some of these other topics, cellular composition. So we talk about omega-3 and omega-6 as we talk about cholesterol. We're talking about increasing the fluidity and rigidity and permeability of a membrane. Things like cholesterol contain functional groups that can influence the membrane of a membrane, the fluidity and permeability and rigidity of a membrane. So, and that allows certain things to occur, certain things through that may or may not be important. So cholesterol increases. The more cholesterol you have, the more membrane rigidity we have, and the more permeability we have. Okay, those are both very important points, membrane composition and how cholesterol affects the membrane rigidity and permeability. Membrane dynamics, lateral fusion, as we've discussed, it's very, very fast. Though this is 1 micron per second. Things just zip around as long as they're functioning laterally. It's uncatalyzed lateral diffusion. However, if we're trying to get from one side to the other, that flip-flop diffusion is very slow. It does not happen very often. It's very, very rare because that polar head group has to get through an entire hydrophobic region, which is not energetically favorable. One important thing that happens in this membrane, as these proteins are kind of floating around, they'll form rafts, which are enriched in sphingolipids and cholesterol. So you can kind of imagine these like groups, and these sphingolipids kind of are more favorable for things like GPI-anchored proteins, caveolin, acyl groups, prenated proteins, doubly acylated proteins. Those are all kind of aggregate in certain regions in what we call rafts. So they have different fatty acid concentration, and they have higher levels of cholesterol, which allows proteins to be there. They aggregate, proteins aggregate in these raft regions, and that provides specific aggregation and functionality at those sites. Caveolin forces membrane curvature. This is very, very common in things like endocytosis. A lot of viruses will take advantage of this. So caveolin forces an inward curvature of the membrane. There's small invaginations. This is a really cool EM of that at the top, where you can see that the cavity of the membrane is kind of dipping in. Caveolin dimers are concentrated in small regions. They kind of force a curvature, and cholesterol molecules, you can see them in there. They kind of interact with that caveolin. Caveolin is a dimer with 6 fatty acyl and 8 cholesterol moieties all attached in there. So you can also have protein interactions. You can have where high density of positive charge on its concave surface interact with the negatively charged phospholipid head groups, and that kind of causes a curvature. You can also have a protein with one or more several amphipathic helices inserted into one leaflet of the membrane, and that crowds the lipids in that leaflet, forcing the membrane to bend. You can also have proteins with bar domains, and that polymerizes into a superstructure, and that will also cause curvature. Curvature is kind of important for fusion. Membranes fuse with each other without losing continuity, so it's almost a seamless process. Fusions can be spontaneous or protein mediated. A lot of times things like viruses will use a protein mediated. Also have release of neurotransmitters that are examples of this and important functionality of membrane fusion. Again, things like budding vesicles, exocytosis, endocytosis, fusion with endosome. Viruses use this all the time. Things like fusion of sperm and egg, fusion of small vacuoles, and separation of 2 plasma membranes and cell division are all examples of membrane fusion. Okay, so now we're going to get into another deep topic, which is transport across membranes. There's 6 ways to do this. Simple diffusion, something goes through. Facilitated diffusion, where we're going down an electrochemical gradient, and so it's facilitated. We can have primary active transport, where something comes in and we use energy to ATP primarily to take against the electrochemical gradient. We can take that molecule and transport it across. We can have secondary active transport against electrochemical gradient, where we take and we move one that's up a gradient and one that's down a gradient. The down a gradient provides energy for the one up a gradient, and that allows for the reaction to be favorable. We have ion channels, where we go down electrochemical gradient, where we just open or close these ion channels. And we also have ionophore mediated ion transport, where we're going down, where an ionophore helps transport an ion across a matrix. Those are the 6 general types of transport across matrix. Things like if I have a polar solute that needs to get across, this is a simple example of how that could occur. I have a transport molecule that decreases, and again an enzyme, so the delta G dagger decreases across that, and so it allows that molecule to go down a gradient. without having a huge activation energy where we go across the membrane. And these can be mediated, and we can turn on and off. Things like CFTRs, which are cystic fibrosing ion channels, are examples of this. There are three classes of transport systems. We have uniport, where we have a molecule and it goes down through. An ion channel, or transport system, I should say, not an ion channel. We have co-transports, where we have two molecules that are transported the same way across, and we have antiport, where one molecule is transported across one way and the other transported across the other. So glucose transport, you can see in the membrane, we have GLUT1, 2, 3, and 4. These are glucose transporters that take the glucose from the bloodstream and put it into the cell. They have actually I should say they have a lot more. They have a lot of transmembrane regions, alpha helices, and those allow for the transmembrane delivery of glucose into the cell. You can look down and see how those residues interface with the membrane. So basically what we have, here's how this works. We have glucose that comes into one conformation. That once that molecule engages in step 2, we get a conformational change from T1 to T2. Opens up. That open up allows the molecule to leave. And then once it leaves, the protein conformation can change back again, and we're back at T1 so that the molecule can then keep transporting down. That's how glucose is transported. Now, having said that, it does need to be turned on. It's not the insulin receptor will turn that on. And there are several mechanisms we'll discuss in biosignaling that will give you an idea of how that works. Bicarbonate, which is a major buffering system in the cell, or inside your body and inside your cell. Chloride bicarbonate exchanger works across the erythrocyte membrane. This cotransport allows the entry of bicarbonate without changing the membrane potential. Basically, we also can use it as CO2 bicarbonate as a CO2 carrying capacity in the blood. And so basically it's an antiporter, meaning the carbonic anhydrase goes one way, and we have the chloride ion going the other way. So chlorine ions are pumped in, bicarbonate is pumped out, and so CO2 comes in, increases carbonic anhydrase due to carbonic anhydrase, and then they get transported out of the cell as chlorine is pumped into the cell. So that allows for maintenance of electrochemical potential across a membrane. Two types of active transport: primary active, meaning that there's an electrochemical gradient, we're pumping out a molecule, ATP is used to pump it out. Or we have secondary active transport, where S1 is pumped out of the cell while S2 is pumped into the cell. So the secondary active transport allows for that to occur. It's against its chemical gradient that provides the energy necessary to perform the active transport. So one of the most cool things that we're going to discuss this semester is this proton pump, the FOF1 ATPase. And this goes across the membrane. It's basically an electric motor it's a proton motor, and it generates energy based on the proton gradient. So it can either make or use ATP. And this is an example of a proton transporter where protons are pumped out and it's used to generate ATP, or vice versa. You can pump protons in and it can use energy. So this is an example of, and we'll see this next week when we talk about glycolysis, actually along electron transport chain. Sorry, not glycolysis. Right before that. We're wrapping up, I promise. Aquaporins are a protein of a tetramer that allows for water to sneak through the cells and to maintain an aqueous balance if we need to. You can see that there's several alpha helices that are formed, and these side chains interact with water and allow it to be chaperoned through. That pore is narrowed down to 2.8 angstroms, which is the size of a water molecule, limiting the passage, but also, importantly, is the charges on these arginine and histidine kind of repel certain things, including the hydronium ion and other molecules. So anything kind of smaller is prevented by the charge, and while the water molecule can sneak through. That's an aquaporin. We also have ion channels. So this is a potassium channel that we're showing here, and you can see the alpha helical structure from the side and from the top. Basically, these chloride ions, the amino acid residues kind of interact with these through the membrane and allow potassium ions that are hydrated to sneak through. And the amino acid residues interact with those ion channels, only allowing that molecule to go through. Really, really cool biological functions that have evolved over time. Gated ion channels, these regulate the transport of ion across the cell. They are usually turned on and off by protein binding. They also respond to things like membrane potential, or and again, the two ways they respond are through binding of a ligand, or if there's a change in the potential. And they have very, very important roles in things like as an acetylcholine receptors, as a glutamate receptor, as a GABA receptor, and a sodium channel, a voltage-gated sodium ion channel. Membranes are electrically polarized, if you haven't figured that out. They're usually -50 to -70 millivolts for the outside compared to the inside, or the inside compared to the outside. Membrane potential is just largely due to the electrogenic sodium potassium ATPase, where we're going to pump 3 sodium out and 2 potassium in. And the flow of these ionic species is very, very important and regulated all the time, not only for the chemical gradient, but also for the electrical gradient. These membranes also allow us to, these electrical potentials allow us to generate energy. And with that, I am going to end the discussion on membranes. Again, the important discussion point today is on the fluid mosaic model. I would encourage you to study that very heavily and understand its role in this process. This topic will be expanded upon quite significantly. All the things we discuss, things like voltage-gated ion channels and the FOF1 ATPase, provide an important foundation for all of the glycolysis and those kind of discussions that we'll be having next week.